Welcome to this session, students. We're diving deep into allelic exclusion, a cornerstone principle of B cell development and immunology. The title, Ensuring Monospecificity in B lymphocytes, tells you exactly what this process achieves. For those pursuing immunology, mastering this genetic lock-and-key mechanism is non-negotiable. Consider the scope of our immune system. It must detect, with perfect accuracy, a single strain of flu virus out of billions of environmental molecules. B cells carry this immense responsibility. Allelic exclusion is the genetic safeguard that makes high-fidelity antibody production possible. We'll explore this crucial differentiation step in the bone marrow, starting from the Pre-B cell stage. Think of it as the ultimate quality control test for every antibody producer in your body. We will meticulously trace the steps of heavy chain first, then light chain selection, and highlight the molecular actors, the RAG enzymes and the signaling receptors that enforce this rule. This knowledge is directly applicable to cutting-edge research and therapeutics, particularly in the realm of targeted drugs like monoclonal antibodies. So, let's turn our attention to the heart of B-cell differentiation. The adaptive immune system is remarkable, capable of recognizing billions of distinct foreign antigens. B cells are the primary agents of this recognition, using the B cell receptor, or BCR. But here is the challenge inherent in our genetics. Every B cell, like every other diploid cell, inherits two complete sets of genes for the BCR, one from the maternal side, one from the paternal side. If both sets, or both alleles, were allowed to produce protein, a single B cell could theoretically display two structurally different receptors, leading to two different antigen specificities. Why is this a problem? Imagine a B cell with one receptor targeting a necessary pathogen and the other targeting a harmless self-protein. This conflict would lead to confusion, inefficiency, and potentially severe autoimmunity. The immune response would be severely compromised by this genetic double-dipping. The elegant solution, which maintains the purity of the immune response, is allelic exclusion. It's a precise genetic gatekeeper ensuring that out of the four potential variable gene alleles, two heavy, two light, only one successful heavy chain and one successful light chain combination is permanently expressed. This mechanism guarantees that the B cell is a highly focused, monoclonal weapon. Before discussing the complex process of exclusion, let's quickly refresh the BCR's architecture. The membrane-bound immunoglobulin is a tetramer, two identical heavy chains and two identical light chains, held together by disulfide bonds. For the heavy chain, the constant region determines the isotype, mu for IgM, gamma for IgG, etc., but the critical variable region is what we focus on today. It determines the antigen binding specificity. The genes for the heavy chain are found on chromosome 14, while the light chains have two options, Kappa on chromosome 2 or Lambda on chromosome 22. Remember a key rule: a B cell will express either Kappa or Lambda as its light chain, but never both. The genetic problem is simple yet profound. Because we are diploid, we have two alleles for the heavy chain locus, two for Kappa and two for Lambda. Allelic exclusion is the sophisticated mechanism that silences the non-chosen allele. By doing so, it ensures that when the cell matures, every single receptor it displays and every antibody it eventually secretes is structurally identical. This homogeneity is the source of the adaptive immune system's characteristic precision and effectiveness. The enormous variability and specificity of the BCR are generated through VD. J recombination. This is the genetic cutting and pasting process that brings together the variable V, diversity, D, and joining, J, segments to create the unique sequence for the heavy chain's variable region and V and J segments for the light chain. The architects of this precise process are the RAG enzymes, recombination activating gene 1 and 2. They are the molecular scissors that recognize recombination signal sequences, RSS, and catalyze the rearrangement. Now, think about the two alleles available. When the RAG complex starts working, it's a random process. It might try one allele and succeed, or it might try and fail, resulting in a non-productive or out-of-frame rearrangement. Allelic exclusion essentially takes explicit and complete control of the RAG enzymes. It acts as a critical regulatory switch. The very moment RAG successfully produces a functional heavy chain or light chain protein from one allele, the switch is flipped, and RAG is stopped from continuing its work on any remaining alleles. This highly regulated cessation of VD J recombination is the genetic mechanism that enforces exclusion. To formalize our understanding, allelic exclusion is the precise molecular process guaranteeing that only one of the two inherited alleles for any immunoglobulin chain is actively and permanently expressed. This is not a passive process of chance. It's an actively enforced, stepwise genetic restriction. We observe a strict hierarchical rule, which is key to its success. First, the cell commits to the heavy chain locus, attempting allele 1 and only moving to allele 2 if the first attempt is non-productive. Once the heavy chain is secured, the same hierarchical logic applies to the light chains. Kappa is attempted first, alleles 1 and 2, and Lambda is attempted only as a final, backup option, alleles 1 and 2. The essential consequence, which is paramount for your understanding of immunology, is that every single mature B cell is monospecific. It has one heavy chain structure and one light chain structure. This means its entire surface and every antibody it will eventually secrete will recognize only one specific epitope. This exquisite and unambiguous specificity is the bedrock of effective humoral immunity. The B cell's developmental journey begins in the bone marrow, starting at the Pre-B cell stage. The initial mission is to generate a functional heavy chain, mu. The cell initiates VHDH-JH rearrangement on its first heavy chain allele. If this attempt is productive, the mu protein is synthesized in the cytoplasm. The cell immediately enters the first critical checkpoint. The mu chain is transported to the surface, where it temporarily partners with invariant proteins called surrogate light chains, Lambda 5 and Pre-B. This assembly forms the Pre-B Cell Receptor, Pre-BCR. The moment the Pre-BCR successfully docks on the cell surface, it sends an immense, life-saving survival and maturation signal internally. This signal is the enforcement mechanism for heavy chain exclusion. It causes immediate transcriptional repression of the RG genes and transiently modifies the chromatin structure around the second heavy chain allele, effectively blocking access to the RAG complex. The cell then commits to rapid proliferation, creating many clones that all share the same successful heavy chain configuration before proceeding to light chain rearrangement. Critically, if the first allele rearrangement was non-productive, the exclusion mechanism does not activate, and the cell is given a second and final chance to try the second heavy chain allele. Failure on both alleles results in mandatory apoptosis. Having successfully passed the heavy chain checkpoint and proliferated, the cell is now a pre-B cell committed to its heavy chain. The next crucial task is to replace the temporary surrogate light chains with a permanent, functional light chain. The process follows a defined, strict order of 4 possible attempts at rearrangement. There is a hierarchy within the light chain loci. First, the cell attempts rearrangement on the first kappa allele, then, if necessary, the second kappa allele. Only if both kappa attempts are non-productive does the cell proceed to the first lambda allele, followed by the second lambda allele. The cell will ultimately express only one type of light chain, either kappa or lambda. When a productive light chain is successfully generated, it pairs with the existing heavy chain to form the complete, surface-expressed IgM B cell receptor, BCR. This functional BCR assembly sends the definitive success signal. This signal ensures final exclusion by triggering the rapid, permanent degradation of the RAG enzymes and a cessation of all further rearrangement attempts on all remaining light chain alleles. The B cell is now terminally committed to its single specificity. Let's solidify the molecular controls that enforce this process. Allelic exclusion is a triumph of molecular regulation. The activity of the RAG enzyme is modulated at 2 distinct levels, the level of transcription and the level of protein stability. When the pre-BCR signals heavy chain success, it leads to the temporary transcriptional downregulation of the RG genes, reducing the number of available recombination enzymes. This is the pause button. When the final BCR signals light chain success, the mechanism becomes permanent. Existing RAG protein is tagged via ubiquitination and sent for complete degradation. This removes the scissors from the nucleus entirely. Secondly, control is achieved through epigenetics, specifically chromatin accessibility. The gene locus currently being rearranged, for example, heavy chain allele 1, is temporarily unwound and open, accessible chromatin. Once a successful rearrangement is achieved and signaled, the remaining unrearranged allele's chromatin is rapidly condensed into a tight, inaccessible state called heterochromatin. This physical modification ensures that even if a stray RAG protein is present, it cannot physically reach the DNA to cause further, unwanted recombination. This tight, dual-level control is what guarantees the exclusivity of the successful allele. Why is this complex process worth the cellular energy? Because monospecificity is vital for a functional adaptive immune system. First, it ensures the purity and efficiency of the response. A B cell must be single-minded and dedicated to one threat. If it had two receptors, the binding energy would be divided, and the resulting immune response would be less potent and less focused. Second, it facilitates high-affinity binding. With thousands of identical, highly specific receptors coating the cell surface, the B cell maximizes its chances of capturing and binding the target epitope, leading to a much faster and stronger clonal expansion. Most critically, monospecificity is essential for preventing autoimmunity, the process known as self-tolerance. During development, B cells are tested against self-antigens in the bone marrow during negative selection. If the single, universal receptor binds to a self-protein, the cell must be deleted or rendered harmless. If the cell had two receptors, one self-reactive, one harmless, the mixed signals would confuse the system, leading to the risk of releasing potentially dangerous, self-reactive B cells into circulation. Allelic exclusion ensures a clear, unambiguous outcome. The cell either passes the tolerance test or is removed. The concept of allelic exclusion is not just theoretical, it forms the core basis for modern biotechnological achievements. Look at the development of monoclonal antibodies, which are the backbone of therapy for various diseases, including rheumatoid arthritis, psoriasis, and many cancers. The entire premise of a monoclonal antibodies is its exquisite, single-target specificity. It targets only one thing. This specificity is simply an in vitro exploitation of the natural monospecificity enforced by allelic exclusion. Hybridoma technology, for example, involves identifying a B cell that makes a single, specific antibody and then fusing it with a cancer cell to create an immortal clone that produces millions of copies of that one desired antibody. This therapeutic power relies entirely on the cell's natural dedication to one specificity. Furthermore, when the molecular controls of allelic exclusion are disrupted due to genetic mutations or epigenetic errors, the resulting B cells may exhibit biallelic expression, leading to disorders. This can manifest as primary immunodeficiencies or lymphoproliferative diseases, highlighting how the immune system rapidly loses its tight control and precision when this fundamental process fails. So, whether we are treating a patient with a targeted anti-cancer drug or studying a rare immune disorder, the principle of allelic exclusion remains fundamentally relevant. To conclude this comprehensive session, let's revisit the core tenets of allelic exclusion. It is the tight, non-negotiable regulation that ensures a B cell expresses only one heavy and one light chain allele. This achievement is orchestrated by a hierarchical series of recombination attempts, heavy chain first, followed sequentially by Kappa and then Lambda light chains. This process is policed by two critical surface receptors. The pre-BCR signals heavy chain success and pauses RAG activity, while the final BCR signals light chain success and permanently degrades the RAG enzyme. The final, critical outcome is B cell monospecificity. This monospecificity is the engine of the adaptive immune response, guaranteeing the purity, specificity, and robust defense mechanism of our immune system. As future immunologists and biologists, understanding this process is essential for appreciating the precision engineered into the adaptive immune system. Thank you for your attention. I will see you for the next class.